said 3 general mechanisms of anemia. This erythropoietic anemia, hemolytic anemias, and post-hemorrhagic anemias. Let us take post-hemorrhagic anemia. Hemorrhage or bleeding, 2 types, acute and chronic. So post-hemorrhagic anemias also could be acute and chronic. About chronic post-hemorrhagic anemia, it leads to iron deficiency anemia because we chronic loss iron. About acute post-hemorrhagic anemia, acute hemorrhage or acute bleeding has 3 stages. First stage is latent stage, when we lose blood, blood cells and plasma equally. So hematocrit will be normal. It's still 45% because we equally lost blood cells and plasma. Second stage is while in case of blood loss we lost blood cells and water, but in this case water is more important for our body than blood cells. So second stage is hydromic stage. We will try to keep water in our body, to retain water, so increasing of production of vasopressin, antidiuretic hormone, and water will shift it from cells and in extracellular space to vessels to retain water inside vessels. In this case, hematocrit will be decreased because we increase water, we increase plasma. Then third stage, our bone marrow will start to produce very frequent, very fast blood cells to maintain cells what we lost. This is third stage. We call this stage bone marrow stage. So 3 stages of acute blood loss: latent stage, hydromic stage, and bone marrow stage. First stage is 1-2 days, second stage 3-4 days, and third stage bone marrow in 6th-7th days after acute bleeding. What hematologist will see under blood film, under blood sample, under microscope, what he will see in case of acute bleeding? He will see, first he will see maturing cells from 5th class in bone marrow. Why? Because our bone marrow become work hard. He want to maintain our cells what we lost. So these maturing cells, they will get out from bone marrow to our blood. So we will see them in blood. Then hematologist will note macrocyte, big red blood cells, because he want to maintain, we need oxygen, understood, due to blood loss. And these macrocytes, while they are big, they will be hypochromic, less colored. So how is the picture, finally, how is the picture of acute blood loss under microscope? We will see maturing cells, acidophilic erythrocytes, polychromatic erythrocytes, and reticulocytes. And we will see macrocytes, and we will see hypochromic erythrocytes. This is the picture of acute blood loss. We didn't say about reticulocyte. Reticulocyte is the last cell from 5th class, and they could be normally in blood about 1 or 2%. We need this reticulocyte to test how is our bone marrow working. If reticulocyte less than 1%, so our bone marrow bad working. Is it normal, 1 to 2%? Normal. If it high, more than 2%, means that bone marrow hard working. So hematologist, he will see also a lot of reticulocyte. Understood? This is the picture of acute blood loss, acute bleeding. This was post-hemorrhagic anemias. What about hemolytic anemias? Hemolytic anemias divided into two types, hereditary and acquired. Hereditary, there are three types: membranopathy, enzymopathy, hemoglobinopathy. Membranopathy, the problem in membrane of erythrocyte. Membrane. The erythrocyte, for example, they are not non-elastic due to hereditary deficit of spectrin. Then this erythrocyte, when it will try to enter inside small vessels, it will lose membrane, and it will take the shape. Shape will be ball shape, as a ball. We call this type of erythrocyte spherocyte, and this type of anemia, spherocytosis. Scientists who discovered this anemia, Mankovsky-Shofar, scientist, understood? Mankovsky-Shofar. This is membranopathy. There are other types of membranopathy except spherocytosis, but they are rare types when the membrane is destroyed, of digmocytosis, for example, also. Second type of hereditary hemolytic anemia is enzymopathy. If you remember, I said about one enzyme inside erythrocyte, it plays role as antioxidant enzyme, G6PD. G6PD. In some hereditary disease, very rare, this enzyme is deficit. So our erythrocytes are non-protected against pro-oxidants. When come some substances which lead for oxidation, our erythrocytes will be damaged. For example, bean product, bean, you know, bean.
Yes, sir.
Yeah. It's more oxidative. So these patients have to have diet to don't eat bean, and to don't take some antibiotics, some drugs, antibiotics. But rare type. Third type of hereditary hemolytic anemia is hemoglobinopathy. Hemoglobinopathy, disorder in hemoglobin. If you remember, I said that hemoglobin consists of globin and hem, and globin alpha beta chains and amino acids. In one case, one amino acid replaced into another amino acid, and we will develop sickle cell anemia. Erythrocyte will have shape sickle cell anemia. Other type of hemoglobinopathy, hereditary, is when no one chain, for example, alpha no or beta no. No alpha, no beta. We call this hemolytic anemia thalassemia. Thalassemia could be alpha thalassemia and beta thalassemia. Understood? Beta thalassemia discovered by scientist Coley. Coley discovered this anemia. How beta thalassemia, how it will be in shape under microscope? We will see as a target board. Target board, you know? Target board. Our erythrocytes will be like this in shape. This is thalassemia. So, if hematologist will see under microscope ball-shaped erythrocytes, he will suggest spherocytosis anemia, hereditary anemia, membranopathy. If he will see sickle shape, so this is sickle cell anemia. If he will see target, so this is thalassemia. This is how it will be under microscope. Hemolysis could be intravascular and intracellular hemolysis. Intravascular means our cells are destroyed inside vessels directly. Intracellular in spleen. In spleen. Which one gives jaundice more? In spleen. Hemolysis in spleen gives jaundice. Understood? Because this in spleen is heavy hemolysis, heavy. Intracellular, sometimes acute, sometimes not acute. This is hereditary hemolytic anemia. Acquired hemolytic anemia, there are three types. Toxic, when some toxins come to our blood and destroy erythrocytes. For example, malaria, and other toxic products. Second type is immune in case of blood transfusion, rhesus factor conflict. Also, it will lead to anemia, hemolytic. This is intravascular hemolytic anemia. And third type is mechanical anemia. Mechanically, erythrocytes are destroyed. For example, in artificial valvular disease, artificial valvular operation, when we transplant valves, artificial valves, and these valves is very hard, and when erythrocytes comes with high pressure, they will be destroyed. This is mechanical. Or if we run without shoes in hard land, and our erythrocytes will be mechanically destroyed. Understood? This is hemolytic anemias. Third types of anemia is this erythropoietic anemias. Do you remember three mechanisms? Yes. I said this erythropoietic anemias, hemolytic anemias, and post-hemorrhagic anemias. We explain this and this. This erythropoietic anemias. It means a problem in bone marrow. First type of anemia, aplastic anemia. Something inhibit bone marrow, all bone marrow. Maybe autoimmune, maybe. And then we will develop pancytopenia. Pancytopenia means all cells low, not only red blood cells, and white blood cells and platelets low. So this patient will develop anemia and will develop immune deficit. It means immune system weak, and they will develop hemorrhage because no platelets, low platelets. Understood? This is aplastic anemia. Other type of this erythropoietic anemia, B12 deficiency anemia. Why B12 lead to this type of anemia, of this erythropoietic? Because we need B12 for DNA. Do you remember I said that for hemopiesis we need DNA, we need growth factors, understood? DNA we received from B12. B12 has two coenzymes, methylcobalamin and desoxymethylcobalamin. Methylcobalamin, what he does, he convert folic acid into tetra hydrofolic acid, and tetrahydrofolic acid we need for formation of DNA. Other coenzyme is desoxymethylcobalamin. What he does, he formates fat acid, what we need for myelin sheet of nervous. Understood? So if no vitamin B12, and the causes of deficit of vitamin B12, it could be no feeding, no input of B12, or no absorption, problem in stomach and intestine, no transport after absorption. We need transcobalamin protein, which transport B12. Or we lose vitamin B12 in breastfeeding and in pregnancy. This is causes of vitamin B12 deficiency. What we will develop? If no DNA, so the hemopiesis will be abnormal, and it will develop megaloblastic type of mitosis. Megaloblastic, understood? So what we will find in blood film, in blood sample, we will find megaloblast and megalocyte cells, very big and hyperchromic cells. They are good colored, hard colored. Understood? And also we will develop pancytopenia. no, it means less of white biopsies and less of platelets, because no DNA, so all three types will be deficient. About fat acids of myelin sheet of nervous also will be defective because no vitamin B12. We will develop paresthesia, difficult disorder in sensation, paresthesia. That's why neurologist they gives vitamin B12 for neurologist patients. Understood? Because other consequences of vitamin B12 deficiency, while there are no DNA, also will suffer other cells which continuously regenerated in our body. We have skin cells, they are continuously regenerated, yes? Skin cells. Mucous membrane cells also continuously. Nails, hair. In case of DNA of B12 deficiency, we will develop atrophic ulceration of skin, stomatitis in oral cavity, gastritis, enteritis because mucous membrane disorder. Then also hair loss and nail deformation, and also taste and nasal disorder. Taste disorder. It will be paro. The patient likes a smell of benzene, for example. He likes to eat chalk, soap. Understood? This abnormal taste and abnormal. This is about B12 deficiency. About iron deficiency, anemia, iron deficiency. You have to know that iron we have where we have iron in our body, in hemoglobin, yes, and in mitochondria, because in mitochondria there are iron containing enzymes. So iron we need also in mitochondria for ATP synthesis. Understood? In case of iron deficiency anemia, what is the causes of deficit of iron? Causes is also no feed, bad absorption, yes, no transferrin absent, or we lost iron. We lost iron by breastfeeding, by pregnancy, and by chronic bleeding. If you remember, I said chronic bleeding. We lost iron also. What we will develop? So no iron in hemoglobin. So anemia. How it will be in shape in blood sample? The hematologist will find microcyte, small erythrocytes, and annulocyte with hole, because no hemoglobin, annulocyte, and hypochromic because less of hemoglobin. Yes, this is what we've heard. So you remember this triad of iron deficiency anemia: microcyte, annulocyte, and hypochromia. This is iron deficiency. In other side, iron, I said it's in mitochondria, so no ATP synthesis. Will suffer also which cells? Which continuously regenerated. Because for regeneration we need energy or not? We need. So no energy, so these cells will be suffered also. Which cells which continuously regenerated? Skin cells, mucous membrane, taste, and hair and nail. So lost hair, nail deformation, skin atrophic ulceration, and stomatitis, gastritis. Understood this? Yes. Yes, there is one last type and very rare type of anemia, very rare type of anemias. When we do blood film and we find microcyte, annulocyte, and hypochromia, we think this is iron deficiency anemia, yes? But when we do blood analysis, we find high level of iron in blood. So this is not iron deficiency anemia, yes? But it seems as iron deficiency anemia. What happened? Do you remember I said that It's coming from glycine, protoporphyrin, coproporphyrin, porphyrin, and then connected iron. It's connecting by enzyme ferrocolitiase. In some hereditary cases, ferrocolitiase is absent. So iron doesn't connect to porphyrin. Then in this body will be accumulation of iron and porphyrin. So iron high in blood, porphyrin localized under skin, causing as a skin rash, and this skin rash is ruptured when he go under sun, and it's painful. And his nail also deformated, hair loss, and teeth also bilateral also deformated, and he feel that he need blood. When he drink blood, he become good. Can you believe? This type of anemia we call vampire anemia. Vampire, yes, drink blood. This general name, but the scientific name is porphyria, because porphyrin accumulated in our body. So, conclusion of our anemias, as you say, that we can, by under microscope, we can suggest which type of anemia developed in the body. Understood? For example, if I found macrocyte, hypochromia, and maturing cells from 5th class, I will think about acute blood loss. Yes. Sickle cell anemia, ball shape, spherocytosis, target board, thalassemia, microcyte, anulocyte, and hypochromia, iron deficiency anemia, megaloblast hyperchromia, B12 deficiency or folic acid deficiency anemia. Understood? These all types of anemia in general. And thank you for your attention.